Okay. All right, great. So welcome. This is the first session of the Tech Forum, so welcome to the Content Standards track. And this is Vishali Patel. I'm a Senior Advisor at the Office of Technology at ONC. And you are all participating in the session that's going to examine addressing barriers to lab interoperability. Next slide. Brett, next slide. Yes, thanks. So before we get started with the panelists and the session formally, I'd like just to take a minute for us to take stock as to where we stand currently with regards to lab interoperability. Back in 2012, ONC supported a national survey of clinical laboratories, which focused on both hospital labs and independent labs that assessed lab capabilities to electronically exchange, looked at the actual levels of exchange activity, the use of standards, and barriers to exchange. We found that about 6 in 10 clinical laboratories nationwide possessed the capability to send results in a structured format electronically to an ordering provider's EHR, and that this varied pretty widely by type of laboratory. Hospital-based clinical laboratories and larger clinical laboratories had significantly higher levels of capability compared to independent and smaller labs. And overall, based on test volume, approximately half of all test results were sent electronically in a structured format to an ordering practitioner's EHR in 2012. However, when we looked at it by lab, about 3 in 10 labs reported sending 75% or more of their test results electronically in a structured format. So although by test volume, over half of lab tests back in 2012 were being electronically sent in a structured format, when we look at it at the individual lab level, a majority of test results were not being sent out in electronic structured format. So there was definitely room for improvement back in 2012. Fast forward 8 years, and we're here in 2020. And back in late December, early January, before COVID-19 pandemic, ONC had decided to support another assessment of lab interoperability. And before proceeding down a particular path in terms of a national survey or other types of measurement, what we wanted to do was take stock of what are the current issues in lab interoperability that warrant measurement, because things hopefully have evolved since 2012, and we wanted to really take stock of what was going on today. And the findings that I will focus on really relate to the content and messaging standards that I'll be presenting on shortly. Next slide. So, great. So with regards to messaging standards, what we identified through doing interviews with key informants, which included representative of lab associations and clinical labs. We had members of our Federal Health IT Advisory Committee who were involved with lab interoperability, SDOs, public health organizations, our federal partners at the CDC, HEAL representatives, and other subject matter experts, as well as a targeted literature review. What we found with regards to messaging standards was that, it would be important to measure 3 key areas, one of which is to determine how many and what types of interfaces labs maintain. Back in 2012, we found that building interfaces to connect with EHRs was identified as a resource-intensive and costly endeavor. And in 2020, our key informant interviews suggested that that still may be an issue and would warrant measurement. Informants noted that labs maintain hundreds and sometimes thousands of HL7 interfaces with ordering providers. However, survey data across a diverse set of labs would provide greater insights into this. A second area that warrants measurement is to determine what factors limit the shift to newer standards, and what would the value proposition need to be to shift to those newer standards. You know, predominantly labs, based on the interviews, are still using HL7 version 2.3.1, and it's working well. However, it would be important to determine what would the new standard, what benefits the new standard would need to confer in order for there to be a shift to that newer standard. And it also relates in some ways to the next topic, which is, you know, is there awareness and interest to use FHIR or other standards to either share test results directly with patients or for other use cases? What we found based on our interviews was that LabCorp and Quest share data with patients via Apple Health Record using FHIR. However, per our interviews, a majority of lab data sharing with patients is primarily done through EHR vendor patient portals. Now, that's not too surprising given the emerging nature of third-party apps. However, with the implementation of the recent ONC regulations, healthcare providers and EHR developers will be making their health information accessible via FHIR APIs. So it'll be important to monitor whether labs will also follow and be making these data available as well. Next slide. And the other key topic that was addressed through these targeted literature review and interviews was terminology standards. And there were three areas there that kind of bubbled up to the top with regards to measurement. One, to what extent are standards being used and for what purpose, and how does this vary across laboratories? Back in 2012, we did ask about the usage of LOINC and SNOMED, but it was measured in a dichotomous kind of way, yes or no, do labs support this? In the future, it would be valuable to measure the volume of test results encoded using LOINC and the observation values encoded using SNOMED to really get the true extent of use. Additionally, it would be of value to measure the uptake of UCOM, a standard for representing measurement units. The second key challenge, the second area that warrants measurement that was identified was how prevalent are challenges to using LOINC and what types of laboratories experience these challenges. Some of the areas that have been identified in the past, based on the 2012 survey, were the time and expertise needed to use LOINC, particularly as it relates to mapping LOINC codes. Now, this would be something that would be worth assessing again to see if that still remains an issue. Other areas that were identified as worth examining include the timely availability of LOINC codes, the complexity and granularity of LOINC codes, which may make it challenging to code consistently, uncertainty about which LOINC codes to use, given that there are many codes that appear to be similar. However, not all the issues that relate to challenges using LOINC actually relate to LOINC itself. For example, the ease of locating a LOINC code and name on standard lab displays and the storage of LOINC codes so users can locate them easily is not really related to LOINC, but to just the user interface. And so that would be another area worth exploring. And finally, an important area to assess would be, and this gets to the topic of our panel today, is what is the level of awareness and usage of tools available to help address some of the challenges? Today we'll be discussing efforts to encode upstream, which could help minimize data mapping challenges downstream, the availability of guidance and resources to using terminology standards, LOINC mapping validation tools to check whether the mappings are appropriate, as well as other areas. So So assessing the usage of tools will be an important aspect of measurement, in addition to assessing the challenges and the extent to which those challenges are experienced by laboratories. Next slide. And finally, I'd like to introduce our speakers for this session. First will be Dr. Michael Waters, who is at the FDA and is the team lead for SHIELD, which stands for Systemic Harmonization and Interoperability Enhancement for Laboratory Data. He'll be describing the SHIELD initiative with us today. And he's also the real-world evidence representative for the Office of In Vitro Diagnostics and Radiological Health at the FDA. Second will be Dr. Clem McDonald, who's the Chief Health Data Standards Officer of the National Library of Medicine. He is a leading figure in the field. He developed the LOINC database, and he is a co-author of the UCOM standard, which I mentioned earlier. And he's one of the founders and authors of the HL7 standard and a major contributor to the genomics reporting chapter of the 2018 V2 laboratory results message. Third, Dr. Stephen Lane will be speaking. He's a practicing primary care physician and clinical informaticist at Sutter Health in Northern California, where he serves as the clinical informatics director for privacy, information security, and interoperability. Dr. Lane wears many hats. He serves on ONC's Health IT Advisory Committee, the Sequoia Project Board of Directors, the HIMSS Interoperability and HIE Committee, the Care Quality Steering Committee, the Da Vinci Project Clinical Advisory Council, and the Direct Trust Clinician Steering Work Group. And he's also a faculty member at UCSF and at an instructor in clinical informatics at Stanford. Thirdly, Dr. Swapna Abhyankar will be speaking. She's the interim director of LOINC and the Health Data Standards at the Regan Street. She's a pediatrician and is also board certified in clinical informatics. And our last speaker is Dr. Ela Singh. She serves as Chief of Laboratory Medicine and Chief of Pathology Informatics at Texas Children's Hospital. And she's also Professor of Pathology and Immunology at Baylor College of Medicine. And she serves as the Program Director for the Fellowship in Clinical Informatics. She's the founder of True Lab, which she'll be talking about today, which is a national initiative to change the names of clinical lab tests, those that are standardized and easily understood by clinicians. So she'll be really providing us, she and Dr. Lane will be providing us with the clinician's perspective on lab nomenclature and today. So with that, I'd like to circle back and ask Dr. Michael Waters to get on the line and begin his talk. And Brett, if you could move the slides, that would be great.
I'm here.
Great.
Okay, so next slide. Oh, there you go. So it really is an honor to get to work with everybody here and help present this a very important message. And what we do within SHIELD, which is a public-private partnership to get interoperability for laboratory data, is take the very keep it simple approach. So I apologize for the very Sesame Street nature of this slide, but we like to just start with the simple understanding of what the challenges are and what we're trying to resolve fundamentally. So here we are just making a quick note that what we're trying to do is make sure that the question and answer that a diagnostic device can provide can ultimately be described the same way every single time, as much as humanly possible. And that means that when a patient comes in with signs and symptoms of COVID-19 or flu or whatever else, that when they get a nasopharyngeal swab specimen collected or whatever other type of specimen gets collected, that gets accurately represented. So when that specimen gets processed and applied to a particular test, that the information that goes along with it and gets populated can be accurately represented. So the question ultimately, after much discussion from a couple of public workshops that were put on as multi-agency workshops and several discussions with stakeholders, the best way to actually represent that question, it was determined to be a LOINC code, and that the answer could most accurately be represented by a SOMA TT code, and a very specific device that was actually doing the question asking of a specimen could be represented by a device identifier. But the fact is there's nothing that's perfect in all the standards that we have, and there's some challenges to actually face with this. But ultimately what we're hoping to do is have that information reported. So this is what we do in SHIELD. And the next slide. The way that we actually try to help that harmonization process is make sure that everybody that is engaged in this entire product process and interactions with diagnostics, that entire life cycle, there's stakeholders that are engaged in the curation of both the device itself and how it actually operates, and then there's the folks that are responsible for the electronic transmission of that information. So what we asked is that could IVD manufacturers actually provide some pre-harmonized coding that was vetted through a sole authoritative source to laboratories so they could decide to use that if it was made available to them. And then that could actually be pushed downstream. And so what we're trying to make sure is the information that gets pumped in in a harmonized way from the point of provenance, so it can actually be utilized in a lossless manner as it moves downstream. Okay, so next slide. So since its inception in around 2012, before SHIELD was actually called SHIELD, we've had a lot of progress, but then come January, our emails start blowing up, and next thing you know, this has taken a very, very critical perspective as far as making sure that we have interoperability for laboratory data. So what you see here is a bunch of links. If you could just go to the next slide, and I'll just note that on these links, there's this right here. They're focusing on the COVID-19 laboratory data reporting guidance, which went live on June 4th. Next slide. And under that guidance, there are a list of required data elements. And if you see that link that says LOINC codes provided by CDC, well, these are actually pre-mapped codes. There's also device identifier. Next slide. There's pre-map codes that are provided in the form of a document called LIVID, which is LOINC for in vitro diagnostics, which also actually includes SNOMED CT codes and UCOM units when quantitative tests come in, which includes how to uniformly describe those laboratory test data elements. Now, it's very obvious here also within the HHS laboratory data reporting guidance. There's a lot of things that don't normally live within an LIS. And these are challenges that we're all trying to work together to figure out how to collate this information together. But SHIELD is focused on the laboratory data side of things. So that means when there's a test order or test result, particularly the test results, actually we're focused mostly on test results. Next slide. What we do is, as laboratories try to get into place with these, you see right here in the bottom of the first paragraph, August 1st, 2020 is when these reporting of these data elements supposed to come to fruition. Next slide. What we've been doing is making sure that as the tests come to market, they get populated in a spreadsheet, which is downloadable through this website that's on CDC. Next slide. And within that document, if you were to open this document up, this is the first slide of, this is the first tab of that spreadsheet that you would see. And next slide. If you click on the bottom tab over there where it says link mapping, what you'll get is a mapping for each of the tests that are currently emergency use authorized to, authorized under emergency use authorization to be used for testing for COVID-19 in particular conditions. And next slide. What you can do when you utilize this slide, the spreadsheet, is you can go ahead and sort through each of these columns. So if you were to sort by manufacturer, next slide, and then model, and then next slide. And even if you want to, you can go ahead and hide particular columns that you're not interested in seeing. Next slide. What you can get to fundamentally is a nice pared down view of exactly what you need to see. So this is an example of what it could ultimately look like. We're always in the process of making sure that we can improve this fundamentally. But what you can see here is you see a manufacturer, a model, a way to actually provide SNOMED CT codes for the specimen type, a vendor result description, so like positive, negative, invalid. Instead of saying positive, you would say detected. Instead of saying negative, you say not detected. And if you want to convey an invalid result, each one of these SNOMED CT codes here are available here. So you could choose what device you have inside your laboratory and represent that. And then of absolutely critical importance here, is a link code that's associated with this to represent that question that is actually being asked. And there's also an order code that's associated with it, and a way to essentially represent a device identifier. If you look over to the right-hand-most side here, what you'll see is a column that says test kit name ID. Hopefully, at the end of the day, we'll be able to have a unique device identifier associated with this, particularly the device identifier component of the unique device identifier. But in lieu of that, just being able to have a name of the test, underscore the manufacturer, is enough to actually get some unique information that's associated with this test. And it's very important to be able to tie these reports within the COVID-19 diagnostic laboratory testing system, the electronic laboratory testing system, so the CELLAR for short. But next slide. If we are to take all this information together and say, okay, well, that's part of the equation, is getting harmonized information that people can out to people so they can use it for reporting purposes. This all ties back to the electronic laboratory reporting guidance. Next slide. And in these links, the first one that's used as a resource here is an example provided by HHS on how to actually encode these different types of information through this specification. And as anybody actually goes through this process and has challenges using this, we would love to know. So if you can actually respond back to us to figure out how we can better iterate towards improvement, this is something we want to make sure that we work with end users on at the end of the day, so we can actually fundamentally achieve laboratory data interoperability in a way that is actually consumable to the greater need for information, which extends beyond laboratory data interoperability. So next slide. So also linked in this HHS laboratory data reporting, this additional guidance that's provided by HHS is a link to how to do HL7V2 messaging for COVID-19 data reporting. And I also encourage folks to go to this website and help get engaged with this group. So next slide. Within this is information on how you can actually do HL7 reporting for each one of the data elements that are associated with the guidance. And next slide. There's also an ability to kind of consume examples within this effort and help give people individualized direction. So we've been doing that a lot for all the different needs that are there across the ecosystem for laboratory data reporting. And there's also resources on where to find this information, so you can get kind of very detailed and directed help that will help you concretely create the build or do whatever you need to do to actually get a success at the end of the day for laboratory data reporting. Next slide. So with that, I just want to say thank you very much for your time, and please don't hesitate to reach out to us. I know there's a lot of questions, and we actively want to help everybody because this pandemic is not going to go away on its own. It's going to go away with all of us helping each other together. So thank you.
Thanks, Mike. And I think Dr. Clem McDonald is on next. Great. Clem, are you on?
Yes, I'm on. Thank you very much.
Wonderful.
And I just want to mention I'm a clinician too and took care of patients for 34 years. And the reason why I got involved at the beginning was as a clinician, it was so darn hard to get lab data in front of my face. But anyway, that's another story. Next slide. You got a lag in the slide. Next slide. Okay, so to set the record straight, the level of granularity in LOINC is criticized, but it's mostly essential. You can't equate a 24-hour urine sodium with a spot urine, nor a glucose reported in mass concentrations with one reported in molar concentration. They yield different numbers, and there's millions of examples like this. The other point is that two labs may choose different codes for the same test and not be wrong. So there are good reasons to have slight difference in specificity between labs. So one lab may distinguish manual and automated differential counts because they report both of them on the same report. And another one, when they don't have to change their steps, and they have to reach the manual test, so they use a non-method specific one. In another case, let's see, I've got to get rid of something on my screen here. Oops, too much. I can't make it go away.
Well, we can still. No, you can keep going though. Yeah.
I got it. Sorry. Oops. No, I didn't get it. I screwed it up. I should never touch the clicker. So LOINC provides equivalence classes and test groups that allow receivers to kind of collapse a similar but similar and clinically similar tests together. And you'll hear more about this from Dr. Abiancar. Next slide. Next. Okay, and now there's lots of usage of LOINC behind healthcare records, just for the record. So for more than a decade, insurance companies have required labs to send, many insurance companies, to send them HL7V2 results with LOINC codes for quality assurance purposes. And that requirement is a source of LOINC codes with lab results in the many large medical claims databases, such as Optum, Proving Health, etc. They're all research databases. NIH has a 1 million patient All of Us research program. There's a big program, N3C, for COVID patients. Odyssey, which is a huge program, international program. PCORNet, which contains 9 billion results, all have LOINC-coded variables and depend on LOINC mappings to combine data from multiple sources. The same is true for health information exchanges. The largest commercial labs, including Quest, LabCorp, ARUP, and Mayo, produce mapping tables from their test batteries or their test catalog to LOINC, and have done so for years. Next slide. we're going to exemplify some content from Mayo. Next slide. So excluding a few cases for which they don't need LOINC code, Mayo Lab has 21,239 test results as of October of last year, which need LOINC codes. They had 97% of them as of last year, and you can see the pictures there. Next slide. So Mayo has done other things with labs. So of the 8,000 orderables in Mayo's lab catalog, they've mapped 61% of them so far and are going strong, trying to finish them all. And of the 4,594 distinct test results that Mayo obtains from more than 80 other laboratories, what a lot of places will call send-outs, the send-out labs have provided lab LOINC codes for 86% of them. So there's a virtuous cycle when one lab asks the other to do LOINCing. Next slide. So PCORNet, I mentioned earlier, is a large collaborative research project with 9 billion variables for more than 60 sources, and they're all mapped to LOINC. So we received data from about 190 million of these map tests from 2 of their sources so far. Now it's done in it's complicated, but we really just we get a sample from which we can reconstruct the numbers. There are 3,600 distinct combinations of LOINC codes, source test name, source unit of measure string, and we used an LOINC mapping validator and manual review to find errors. And the weighted mapping error rate in the sample is less than 2%, but we can't trust that until we look at all the data. Next slide. Now, the errors we did find, and this is encouraging, they're mostly confusion between pairs of choices, molar concentrations versus mass concentrations, spot urines versus 24-hour urines, absolute counts of cells versus percent counts, mass concentrations such as versus mass fractions, such as the percent O2 that's oxygenated, tests with positive, negative versus tests reported as a concentration of arbitrary units. So the right choices of these scales can be easily identified as the units of measure, and we urge people to look hard at the units they have in the test before they complete the mapping. Next slide. We saw very little evidence of mapping errors due to the number of LOINC codes, nor its complexity. One exception might be urinalysis, which is relatively messy because there's the electronic machines can report out the same data 3 different ways. There's 2 kinds of electronic reporting, and there's all the manual ways in between. So that was the reported local test names abbreviations for non-routine tests are also very short and often too short to decipher. Longer names would be more important than perfect names to be more complete in what they mean. Now, labs themselves have background data, so they don't have as much problem. But if a receiver tries to map, they're going to have real problems with the current names. Next slide. So the LOINC mapping validator, it's not cooked completely yet. So I wouldn't count on it tomorrow, but soon. So the first version had 7% false positive rates, which that is they identified correct mappings as errors. We now have it down to 4% false positives, but we have more work to do. And so check back on the URL in a month and think we'll have it down to 1%. And also believe for many incorrect mappings, the program could actually suggest what the right mapping is because of this pairwise relationship between the erroneous ones and the right one. And this is the URL, but again, don't trust it yet. Next slide. UCOM and the LOINC validator. So the UCOM is what the validator depends upon. And the UCOM process will do some guesses, mostly right, about what the real UCOM codes are. So it'd really be important to have good UCOM codes and messages. And remember, that doesn't mean you can't also send your routine units strings, whatever they are. So we have a web-based and an API NLM-based UCOM validator, which if people want to make sure their UCOMs are right, you can check it on the URL. I think we've got about 300,000 uses a month so far on that. Next slide. The mapping burden can be reduced. So firstly, those who are mapping start with the use of the 2,000 most common LOINC subset instead of having to dig through the 44 some odd thousand there are. And Dr. Arbianca will describe a new web browser from LOINC, from RegUseek, that will do this. Then also take advantage of the LOINC mappings from your referral labs. An awful lot of institutions do send out most of the oddball tests to referral labs or commercial labs. And then if they don't give them to you already, the big ones do push them to provide such mappings and then push your institution to take them in instead of throwing them away. And they also take advantage of the mappings between internal instrument codes and LOINC, the IVD manufacturer support. Now, this is something Michael talked of that he didn't emphasize. Libid has mappings between the instrument, the codes put out by the instrument, and LOINC codes from at least a number of large IVD vendors, including bioMilieu, Abbott, and others. So look for those, and those who don't do it, push them to do so. And then once we get it really good, use the validation program to find errors and take the corrections it suggests. But it'll take a month or two before it becomes good enough for prime time. Next slide. Okay, why LOINC is needed. All tests don't come from the same source, and it's getting more and more that way. If clinician's office practice, he may have different labs used for different patients because they have different insurance companies. And we need common identifiers to tie them together. In some settings, the source lab changes with the patient's change in insurance. In most hospitals, a major share of non-routine tests are send-outs. LOINC provides a way to integrate those into the local labs and EHRs. Health information exchanges contain labs from many sources and need a lingua franca to file them sensibly. Next slide. So LOINC errors, never mind, skip that. Oh, yeah, the one thing, most EHRs report their local test names on their screens and in their reports under their local names, not the LOINC code name. So there's a little chance that the LOINC codes being different caused the problem to clinician. However, next slide. The fact that there's not visibility of LOINC in many EHRs is a problem. LOINC codes are rarely stored as part of the test result in EHRs as they are within the native FHIR and V2 messages. Researchers tell me they can't even find the LOINC codes that they know are for sure somewhere in their EHRs. They may require digging through a thicket of internal mapping tables to find them. Apple Health is the major exception. The standard codes and data structures can be reviewed via a mouse click. Next slide. Next slide, okay. So 1,000 eyes will find all problems. That's sort of the mantra of the open source movement. When everything is visible, problems are detected and quickly fixed. The near invisibility of LOINC codes hides problems such as mapping errors, so no eyes see them. The whole process would improve if they were more visible and kept in the record together with all the results the way they are delivered. And if users and testers and managers could click on something to see what they really are when they're looking at new medical record results. Next slide. Okay, that's all I've got, and thank you for listening, and we look forward to questions later.
Thanks, Dr. McDonald. We have Dr. Stephen Lane up next.
Thanks, Vaishali, and thank you for inviting me to participate in this panel. It's really an amazing collection of experts who are really doing the deep work to solve the problem of lab interoperability. Next slide. And we can go on one beyond down as well. So I'm going to talk a little bit, provide a little bit of background, talk about the work that we did on the ONC Interoperability Standards Priorities Task Force and the recommendations we made related to lab interoperability, talk a bit about work we've been doing within the Care Quality Commonwealth Joint Document Content Work Group, and then sort of looking ahead at new opportunities to support lab interoperability. On the next slide, Vaishali kind of went through my background, but just to be clear, The speakers that have been presenting thus far are really deep in the weeds of trying to fix this. And what I want to do is just back it up and give a little bit of a clinician's perspective. I've been a practicing clinician on an EHR for 3 decades, informaticist, and focusing in on interoperability for quite some time. As Vice Charlie mentioned, I'm involved in a number of different initiatives, most of which are looking at this problem and trying to solve the problem. This is so important, getting lab data to the right user at the right time in the right format with all the data normalized so it can really be useful is really a bit of a holy grail. So it's something that is worth your attention. Next slide. COVID certainly has pointed out very clearly some of the challenges. Somewhat unique, really. A lot of these are new or different, and so we're right in the thick of this, where you've got testing going on at all sorts of sites, new and changing laboratory tests and methods. The data standards that we're discussing today are evolving, in front of us. We've also had challenge with delayed results, patients changing care setting by the time the results get back, repeated testing, which is very appropriate in this context. So there are a lot of opportunities for us to use COVID-19 as a test bed to try to get things into place quickly that we've been really trying to work on for some time. Next slide. So I have had the opportunity to work with the ONC and to co-chair, along with Ken Kawamoto, the Interoperability Standards Priorities Task Force. This is workforce worked together through 2018 and 2019 with the charge of making recommendations on priority uses of health IT and the associated standards that support such uses. We published a final report at the end of last year. I included the link there. And the first order of business for this group was looking at orders and results and how to make that data more interoperable. On the next slide, we made some key observations about lab orders and results, many of which have already been addressed by some of the speakers and will be addressed further, but clearly the need for consistent encoding of orders and results and values so that that data can interoperate meaningfully between systems. We acknowledge the challenge that Clem was talking about granularity of codes, and it's absolutely true. You need a different level of granularity depending on your use case. But it's very clear that we need standards and consensus across the industry so that the information models, metadata, and terminologies can be translated between systems as the data moves. There was discussion already about orderables being standardized, very important, because really you want an order to be interoperable as well. A patient gets an order, they should be able to take it where it needs to go, find the most efficient, highest quality, most available testing site. We're nowhere near that. Results, of course, need to be available for patients and their proxies if we want to support patient-centric care. And the team really identified the need for unique reference IDs for result data so that those can be deduplicated when they're coming at you from various sources and versions can be accommodated. Next slide. So out of this work, which spanned a period of months, we came out with some key recommendations that resulting agencies, be they the big labs or individual providers, should consistently provide LOINC codes and SNOMED CT to encode the observations. That the level of granularity problem really needs to be addressed. And just as Mayo is doing, when you need greater granularity, go get it. Work with LOINC and then be sure that that's encoded into the data stream. That the metadata, including the methodology, the units, the reference range, using UCOM, for example, flags for abnormals, all of that needs to flow to the clinicians who ordered the tests and anybody else who ends up getting those results after the fact. That data needs to be included, the data is transmitted between systems, as well as when it's maintained in EHRs and other HIT systems. As Clem was saying, oftentimes it's there, hidden away somewhere, or it isn't there at all, and it really needs to be maintained so that it can be transmitted. So that was just a very brief summary of what we did at ISPTF, and I invite you to review the final report. On the next slide, I'm going to talk a little bit about the work we've been doing within the Care Quality Commonwealth Document Content Work Group, which we chartered in 2017. There were independent groups working within the Commonwealth network and the Care Quality Framework to try to look at document content optimization. So we came together and worked on a first publication about encounter summary CDA documents with clinical notes, very important. And then we've been working on a few other projects. But one of the desires that we've had has been to work on just this problem of discrete computable lab result data that can be exchanged between vendor systems as part of query-based document exchange, as well as other methods of interoperability. So in April, as the pandemic was ramping up, we decided to spend some time on this. We had a brief period of time and some energy to expend, and we did that. And on the next slide, we did try to talk about the problem, try to identify what the issue is. That a lot of tests have been created to address COVID testing needs. There are novel test types that the vendors have had a hard time consistently incorporating this discrete codified test data into CDA documents that they can then share between different partners. The structure of the data did not lend itself to translating from the V2 messages received into CDA. But the good news is a lot of work has been done indexing the test types and the result values, and this is now available. So the goal in this work was to try to bring EHR vendors and other health IT vendors together to work on how to incorporate this additional information into the data stream. On the next slide, we identified a number of pathways towards a solution, some of which sound familiar: standardizing the data that's going from the labs to the EHRs in their V2 messages, supporting the mapping of non-standard codes to standard codes, all of these, the great work that's been done by Loink, Livid, Shield, et cetera, that we've been discussing, making sure that that is being utilized, and then in time to harmonize the results in the CDA documents and to automate this process so that vendors can get this up and running quickly. On the next slide, I'm just going to talk a little bit about some new opportunities that we see here to improve the transmissibility of lab orders and results between systems. As you've heard, most of orders and results flow across V2 interfaces, which are tried and true and have been optimized for years, but they take a lot of time and some resources to stand them up and keep them up and running, and they're really just point to point. What we need is a more flexible interoperability platform that can be used, and there are a number of opportunities that we have today to do that. Earlier in the sessions today, there were this discussion about direct interoperability. This can be used as an alternate transport mechanism to push lab orders or results between stakeholders. This is readily available technology that's supported by all certified EHRs. It's got high security built in, and it's a low cost to implement and maintain. So for labs, it would be very advantageous for labs to be able to receive test orders via direct. That would get us away from this reliance on the point-to-point V2 interfaces. And then for those labs to send the results down to the ordering providers, CC providers, and others who need that data after the fact, the patients, the HIEs, public health departments, et cetera. Another option is to send the results via the tried and true V2 method from the lab to a technology partner who can then normalize data from multiple sources and then distribute that out to those who need it using direct. And I've just put in the logos of a couple of companies that are working on just that. On the next slide, which I think is my last slide, as you heard, I'm also involved with the Care Quality. Care Quality, of course, is a partner to the development of the Trusted Exchange Framework that is being supported by ONC. Care Quality also can be used to help support the interoperability of lab result data. The idea here is that providers or other stakeholders with a need to know could query performing labs or others who hold this data and receive that data quickly. This would avoid the need to build and maintain these V2 integrations and really enable all providers to get access to the data that they need. Today, if you're on the care quality framework through one of the various participating networks like Commonwealth, eHealth Exchange, et cetera, you have the ability to query other provider organizations and download results. In most cases, that download is really view only. There are only certain vendors who figured out how to map those results. So when you query for them, they actually come down in a computable format that you can then naturally integrate into decision support, analytics, et cetera. So the goal here is really to try to make this data more available for query, whether it's via CDA today or Firebase query in the future. And again, I think all the work that people are doing with the coding, the metadata, et cetera, is going to support these new interoperability tools as we try to move this data more easily across the ecosystem. And that was my last slide.
Great. Thank you, Dr. Stephen Lane. Next up, we have Dr. Swapna Abhyankar from the Regenstrief Institute.
Great. Hi, this is Swapna, and thank you for having me. Next slide, please. So today I'd like to focus mostly on resources. So, I think it's a common thing that people bring up where there's too many link codes, or, it seems like there's too many link codes and that we need a way to figure out how to choose the correct code and how to aggregate the data. And so I'd like to focus on some resources and tools that we have for doing just that. Next slide, please. So I guess just to start, as just a little bit of background, I just wanted to clarify that all link codes are created based on user requests. We have a lot of codes, but somebody requested them and is using them in some context. And when there are gaps, that's usually because nobody has requested the term yet. So, I would ask that if you're, looking for a particular link code and there's a term that seems to be missing, please go ahead and request it because we're still creating new codes. And then the other thing is that, we get a lot of requests, and sometimes we're not sure if we necessarily need to make a new code or not because maybe it is too specific or too general, and so we actually get the input from the LabLink committee on those types of requests. And so I just, I want to clarify that we don't just sort of make codes in a vacuum. We make them because they're codes that people need for their particular purpose. So I wanted to, sort of go off of what Clem and what Mike Waters presented a little while ago and just point out that IVD manufacturers have been providing link codes for their test kits, and a lot of the major manufacturers have been putting out their mappings in this livid format. And we've been working with a lot of IVD manufacturers as well to help assign link codes and to help them out with how to provide the data in this format as well. And some of these are publicly available, and then some are kind of like behind a firewall or behind a login and are reserved for their customers. But these resources are available out there. Next slide, please. And the other thing that Clem mentioned was that a lot of the reference labs have link mappings already, and so I would definitely take advantage of them. And so I just have some screenshots from some of the big ones. I think they're probably the same ones that he mentioned. Mayo, ARUP, LabCorp, and Quest. And basically, if you go to the specific website and look for the test and you go to that web page, you'll see the link codes mostly for results, but there are a couple that also have the links for the orders as well on those pages. And we work closely with the labs to help them assign those as well. One caveat, though, so, every test kit can be used sometimes slightly, well, okay, not every test kit. Some test kits can be used different ways, and some labs will implement them in, slightly different ways. And so you definitely want to double check on the link mapping just to make sure it makes sense. So, for example, if one particular kit can be used to report quantitative results, or the interpretation of that quantitative result is a, qualitative, detected, not detected, you want to make sure that the code that you pick aligns with your use of that particular test kit. Next slide, please. So one resource that I think is really important is a whole series of guides that we're working on for choosing the correct link term for different domains in the laboratory space. And so this is basically a multi-year effort. We started with microbiology a couple of years ago, and now are in the process of working through allergy and chemistry and drug tox and a whole bunch of others. And the work is expected to be completed over the next year or so. And basically what we have is a process where, we write the draft, we have a stakeholder review that's public, then we have pilot testing, then we have a follow-up second stakeholder review, followed by publication. And so each guide basically goes through all of those different steps. So currently, the allergy guide is in the review phase, and chemistry and drug tox are in the pilot phase, and serology, hematology, and molecular pathology are coming down the road in the next several months. I also want to point out that there's a quick start guide, which is basically an introductory guide that's supposed to be used as a companion to all of these other guides, and that basically provides a very high-level, overview of how to choose the right link term based on, component and property and units of measure and all of that. And so hopefully that, all of these guides will be useful to people when they're trying to pick the right link codes. And we are currently looking for reviewers and pilot sites for the allergy guide. The first review period for allergy ends on August 14th, so that's this Friday. But then we're still looking for pilot sites, and then the second review period will open up probably in September or so. And then I expect the final review for chemistry and drug tox to be opening up in the next couple of months as well. Next slide, please. So another resource that I'm really excited about is the Link Knowledge Base, and this is something that we launched just a couple months ago with the June 2020 release. And this is basically on our website, and it's basically a searchable format for a lot of resources that we previously just had as, PDFs or isolated in their own spaces. And so that includes the Link User's Guide, the RAMA Manual, frequently asked questions, the Link license, and we're hoping that over time we'll be able to include, all of the release notes, all of the release and README notes that are associated with all the different artifacts, I think, that are approaching about 20. And so I think it's going to be a very valuable resource, but even just having the Link User's Guide online, I think is huge, because I personally probably have at least 20 copies of, the Link User's Guide in PDF form on my computer, but now that it's just in the knowledge base, it's a much easier resource to search. Next slide, please. Another resource that we have is descriptions, and so all of the terms that we've been creating, over the last several years at least, nearly all of them have either a part description or a term description, and even though these descriptions aren't necessarily, definitive, they don't help define the link term, but they do help users choose which term, might be appropriate for their context. And so these are just some screenshots. Next slide, please. And then the new search link, which is coming soon, and Clem mentioned this. Unfortunately, I don't have any screenshots because it's literally, we just launched this a couple of weeks ago, but I'm super excited. So I think a lot of people on the call have probably seen, the current search link interface, which is basically, you see the box, you type in your query, and you get the search results. And we have a lot of different, things for how you can search for different codes, but if you don't know the syntax, then you're not going to be able to use them. And, we have the syntax documented, but I think it's difficult, especially these days, because so many of the web interfaces are, you see all the controls right on the screen in terms of filters and things. So I think it's hard when you come to a website and you don't see any of that. It's hard to know, like what filters you can use. So we're really excited about this. We should, have something in the next couple of months, at least for searching LOINC terms, but then eventually it's going to include searches for parts and answer lists and groups in the, very near future. And imagine something sort of like I don't want to mention any big names, but, like major websites where you see the list of filters down the left-hand side and they're dynamic in terms of, changing with whatever your query is. And so those filters will include things like, the property timing, all the major parts of the LOINC term, but then also things like, is this part of the top 2000, or, is this a deprecated code or an active code? And then related to the top 2000, we're going to have that filter, but what we really would like to do is gather new data about the top 2000, because the current top 2000 list is based on data that's, at least 10 to 15 years old. And so if anyone is willing to provide new data to refresh that list, because especially things like molecular tests, there's not too many in there, that would be great, and you can contact me directly. Next slide, please. All right, so then in terms of aggregating the data, we have a couple of different tools that are available. One is the hierarchies, and so we basically have hierarchies for individual parts of the LOINC term, including the component, system, methods, and classes, and then we have a multi-axial hierarchy, which is actually based primarily on the component, but then also it includes the class and the system or the specimen. And these hierarchies are available for viewing in Realma, and I have a screenshot. Sorry, I realize it's probably not very clear. But the multi-axial hierarchy is also available as a computable CSV file. And so, I think this is one of those sort of hidden resources that we have that a lot of people don't know about. And what we would like to do is expose them better, and especially, the individual hierarchy. So, if people have ideas on how best to do that and how to make this information available, we would love to hear from you and work with you. The other thing is that we've been working to map the LOINC parts, especially the component parts, to external ontologies in order to make our terms, and their hierarchies more computable. And so far we've had efforts to map to KEBI and PubChem, the NCBI gene taxonomy, and lots of different ontologies as well. Next slide, please. And then LOINC groups. So this is basically an effort to create sets of LOINC codes that can be used for different purposes, where, such as research or for flow sheet use and things like that. And this is a big area that could, have a whole talk by itself, so I'm not going to spend much time on it, but you can go to loinc.org/groups if you want more information. And I realize I only have 1 minute left, so I'm going to go really quickly just through COVID. So next slide, please. So just a couple of things about COVID. So, we have a lot of LOINC terms related to COVID-19, both lab testing and case reporting and things like that. And all of these terms were created, again, in collaboration with IVD manufacturers, CDC, FDA, APHL, and lots of others. And we've been turning these around, just within a couple of days. Next slide. We have a couple of different websites. One is an FAQ page that helps you choose the correct LOINC term. The other one supports downloads of filtered subsets of these codes. Next slide, please. And actually, you can skip this one. This is just a Livid screenshot that Mike had shown. So I just wanted to show one last thing. So this is the component hierarchy specifically related to SARS-CoV-2. And so this is what I was talking about a minute ago, where you see, coronavirus, SARS-related coronavirus, but then you see the hierarchy under SARS-CoV-2 of the antibodies, the antigen, the RNA, et cetera. So I just wanted to point out that, we do have a hierarchy, and this is computable. And if people have questions, they can contact me. And I know I'm out of time, so I will stop there. Thank you.
Okay, great. Thank you, Swapna. And our last speaker for today is Dr. Ela Singh, who will be talking about the True Lab Initiative. And after her presentation, will be, I've been scanning the chat. I'd encourage the audience members to submit your questions. I've been reviewing them, and we'll tee up with a discussion following Dr. Ela Singh's presentation.
Thank you so much for this honor. I'm going to talk about True Lab, which is an initiative to ensure that lab tests are ordered correctly. The next slide shows that we had a case of measles at Texas Children's Hospital, and nobody could find the test for measles, and there was even talk about sending it to the CDC for testing. Eventually, it was found under Rubiola IgM. Measles has become rare enough that people have forgotten that the virus that causes it is Rubiola. The next slide. The objectives for today is that I want to help recognize that names of lab tests can lead to errors in ordering and serious patient safety concerns. Recognize that many lab test utilization management programs utilize lab test name change as a major tool. And I'd like us to all engage with a process to create lab test names that are easy to understand, use, and make widely available. The next slide goes into why there is an uncertainty in ordering of lab tests. So U.S. primary care physicians are uncertain about which test to order 15% of the time, and 8% of the time they're uncertain about interpreting the results. Next slide. Why is there this uncertainty? Because there are over 4,000 lab tests. Lab medicine teaching hours in medical schools have been reduced often to less than zero, and then test names are confusing. Next, please. The first scenario that I'm going to discuss is where test names are well known, but there's a lack of standardization and clarity. The next slide shows the well-known hemoglobin A1C, which can be called glycosylated hemoglobin, glycated hemoglobin, HbA1c, HbA1c, or just A1c. They all start with different letters, makes it hard to find the test. The next slide shows a set of panels where there's no standardization at all. Liver function panel could include any number of tests. And there are hundreds of respiratory virus panels, and the components depend on the manufacturer that designs the panel. Someday, this particular problem will have a technical fix. So you can hover over the name, and it'll explode to its components. But that's not there yet. Next slide. We're going to talk about a second scenario where the test names are difficult. The next slide shows a well-understood example, vitamin D testing. There are two forms of vitamin D in the body, 25-hydroxy-vitamin D, which is the best indicator for vitamin D status in routine screening for deficiency, or the 125-dihydroxy-vitamin D, which is rarely needed as a test. It's misleading in screening for deficiency and is usually much more expensive. The next few slides, when I talk about the best screening test, it'll be in blue, and the 125, the wrong test, will be in red. Next slide. Talk about 3 hospitals which face the same problem. The wrong vitamin D test was ordered over 30% of the time, and they came up with 3 different solutions. The first solution is on the next slide, where somebody in the lab would call the ordering clinician to find out what their intent was. And you can see the red bars, which show the number of times the wrong test was ordered before the intervention was, about 30%. During the intervention, which lasted about 2 years, the red bars became very small. And then when the intervention was stopped, the red bars became taller again. So people went back to some extent to the original way of ordering. The next slide shows solution number 2, where this hospital decided to change the names of the tests in the CPOE. They took the right test and they called it vitamin D for deficiency screen. The wrong test was called vitamin D for bone and renal disorder. Paradoxically, the wrong test went up. People started ordering the wrong one even more frequently. And this was because a lot of people who identified themselves as bone and renal experts thought that this was their test. It had their name on it. So the solution this hospital took was to completely remove this test from the CPOE. The third option was what we went with, which was, in the next slide, just providing clarifications to existing names without completely changing them. So to the 25-hydroxy, we just appended for deficiency screening, and to the wrong one, we said not for deficiency screening. The next slide shows what happened. So before the intervention, so the red line shows the urinalysis orders throughout this period of time. Before the intervention is on the left, where you can see that the ratio between the right test and the wrong test was 6.5. After the intervention, the ratio went to 30:1, and it has stabilized at this level. The next slide shows that there are several tests that have confusing names. Lupus anticoagulant may or may not have anything to do with SLE, and in the body behaves as a procoagulant. We just talked about measles. I have received a phone call from a clinician asking if the right test for herpes simplex virus was the one that was labeled HSV 1/2. There's also confusion between the epidermal growth factor receptor and the estimated glomerular filtration rate. The two are unrelated, obviously. But many EMRs convert all test names to uppercase. Testosterone-free dialysis by liquid chromatography tandem mass spec is the preferred test to order free testosterone, yet many clinicians won't order it because the word dialysis, which is meant to indicate how free testosterone is separated by bound testosterone, basically it's a method, they interpret that as, well, my patient's not on dialysis, so I shouldn't get this test. I should get the one that's 12 times more expensive and has the word bio available on it. Again, tests which have the words comprehensive or whole are much more preferred than more targeted, more appropriate tests. Again, another example from our own EMR, lymphoma leukemia flow cytometry is really hard to find. And it was a holdover from the days when we were limited by 18 characters in our test display name. Brand names have their own problem. Hardly anybody, except people in that field, realize that quantiferon gold test for TB is an interferon gamma release assay. And so if you were to change quantiferon gold to another company, it would be hard to educate people what they were getting. And at least one clinic has been ordering the free PSA instead of the much cheaper regular PSA because they thought that that test was free of charge. Next slide, please. So the problem is because, we make these names without consulting with folks in other institutions, without a style guide, and without consulting with clinicians who use these tests. The next slide shows the current method that exists in most institutions to fix this problem, which is it goes to a committee which comes up with a solution, and then it takes many months to implement that solution. And there's significant safety challenges in doing that. The next slide shows that there are previous attempts in renaming tests. So there's the CLIC CDC attempt, there's the ONC Tiger Team, Standards for Pathology Informatics in Australia, the Canadian PCLOCT, and then LOINC, which is great for machine interoperability, but unfortunately neither the short nor the long LOINC names were intended to be used as display names. The next slide Actually, we can skip that one. Let's just go to the next one. The next one, please. Next slide. So TrueLab is test renaming for understanding and utilization, and our goals are to create a consensus guideline, consensus names, and to promote the adoption and implementation of these names and guidelines. The next slide shows our many members, and these are many professional organizations at the national level. Many of them are pathology organizations. AAFP is Association of Family Practitioners. There are several EMR, LIS, and terminology groups. There are reference labs involved in this, as well as instrumentation makers. We have federal liaisons, the CDC, the FDA, and most recently the ONC. And we have international partners, the National Health Service in the UK, the Standardization Group in Norway. And we are all working together much more before COVID-19, but we are now getting back to the arena again. The next slide shows some of the work of, so we took a survey done by ASCP and TrueLab to identify some of these problematic names. And the next slide shows the attempt to use existing guidelines to take a name and see what different guidelines would call that same exact test. So you can see that Brian Jackson was the one who assigned us this list, and just say, for example, the LOINC name was chosen by Dr. Swapna Bhayankar, who you just heard. So the next slide shows an example of such an exercise where Seattle Children's Test for Thalassemia Screen was named with different guidelines, and the LOINC name was hemoglobin A2 blood mass fraction. The Canadian guidelines called it hemoglobin electrophoresis panel. You can see the differences here. And the next slide shows a similar exercise with the HIV-1 quantitative real-time PCR name given by Quest. And partners, Massachusetts General Hospital guidelines would call it HIV viral load by PCR. ARUP would call it very similar to Quest, but instead of real-time, they call it reverse transcriptase PCR. LOINC has HIV-1 RNA NAA plus probe, log number over volume. Fortunately, very few clinicians know that NAA is an acronym for nucleic acid amplification. So the next slide, please. So this is going to be an iterative process. We will, design better names, apply new guidelines, use surveys, test, implement on a small scale, finally disseminate for widespread adoption, and ultimately we hope that the standardized display names, order names, will be part of the foundation build of EMRs, LISs, and they will be built to be clinician-friendly, easily understood. So the next slide, some of these names are going to be easy. So measles, you can just say rubiola IgM for measles, and that would be clear enough. But then there are names like the next slide shows, the Noonan and Comprehensive Rhasopathies panel, which interrogates all of these different genes that are mentioned here for their mutations, which are involved in syndromes below. Try putting that on a simple test name. It's not going to be easy. We realize that. My last slide is a call for everyone to participate in TrueLab. Please join us. Help us recruit more clinician organizations and instrumentation makers. And thank you very much for listening.
Great. Thank you, everybody. Thanks all the panelists. And we have some time for some discussion and Q&A. And questions have been coming in through the chat, and I will start with a question for Dr. Stephen Lane, and others can address this as well. Has the group considered defining a content profile or implementation guide specific to lab results, where the content is a FHIR bundle so that it can traverse all of the existing exchanges, meaning the document exchanges do not CDA, they can transport a FHIR bundle as they are content agnostic. And then they go on to say a FHIR bundle may be easier to create and consume, and one can leverage modern edge systems while leveraging existing exchanges such as Direct Project, Commonwealth, Care Quality, eHealth Exchange, etc.
Yeah, that's a great question. And I am not deeply involved in that work within the FHIR community. I think that it clearly was discussed in our Interoperability Standards Priorities Task Force, the need for both FHIR and CDA-based exchange methodologies that would include all the relevant data and metadata. It sounds like the questioner is pretty well versed in FHIR, and I think if there is an opportunity within the FHIR community to move that forward, I certainly encourage you to take that opportunity and get involved in it. But I don't know the details. Other presenters may know more about that.
Okay, great. Thanks, Stephen. This is a question for, I think, Clem and Swapna. One question is, given that different LOINC codes are needed and appropriate for labs, what do you think about the LOINC Top 2000 and the encouragement by ONC that these codes be the codes used in exchange messages? Note that they are required. Note that they are not required, sorry, but encouraged. So I don't know if Clem and or Swapna would like to comment on that. Obviously, Swapna, you spoke specifically about this, so I don't know if you want to take it first, and then Clem, maybe you could add to that.
Sure. I think it's good to have a set of codes to start with, especially if you're not quite sure where to start with choosing LOINC codes. But on the other hand, I would hate to restrict just to the top 2,000 because, as we've seen, there's tons of examples of lots and lots of specialized tests and different use cases for having more specific codes. So I think it's a good place to start, but I certainly wouldn't limit to that set. And like I said, the existing set of top 2,000 are several years old, and so I certainly wouldn't limit to that particular set of top 2,000.
Got it. And a question, I don't know, Clem, if you wanted to add anything to that. If not, I will move on to the next question, which is for Dr. Ila Singh. Is there a plan for a central source for the TrueLab display name result work to be housed or distributed? And they go on to say, using LOINC to house or distribute that information would be a logical choice as that contains the rest of the information.
Yeah, great
Ila,
question.
I don't know if you want to come.
Yeah, sure. Great question. We are a few steps away from that. One of the things I want to emphasize is that when these names will be created, there will be ample testing with clinicians to see that they actually work well, that there isn't a confusion, there isn't, because that's one of the things that usually the way we name our tests is we don't ask for feedback from people who actually use those names. So before COVID happened, we were getting ready to do some of those surveys, and we'll now get back to doing that. And once we hear from those surveys, that'll help us, fine-tune those names So we are a few steps away from creating this wonderful list for 4,000 names that can be made public.
Okay, great. Thanks, Dr. Singh. Another question for Swapna and Clem. I think he may have lost his connection and may be getting on. So maybe, Swapna, you could address this point. This is an issue that one of our audience members brought up for you to comment on. They say, I suspect errors arise from sites not maintaining their maps after implementation. When a test name, unit of measure, specimen, etc. are edited in the lab's catalog, the LOINC mapping needs to be reviewed. I know both you and Clem, talked about different tools related to the LOINC mapping, and I was wondering if you could address the whole issue of like maintaining the LOINC codes and strategies around that.
Yeah, so we have a recommendation actually that LOINC is published twice a year in June and December, and we have a recommendation that organizations should update their LOINC catalogs within 90 days of each publication. And, sometimes that seems a little bit daunting, but there's actually really easy ways to figure out which new codes there are or figure out which codes have changed. And so, yeah, I think that's really important because, I've seen lots of cases where somebody picked a LOINC code 6 years ago and then never updated it, but there's, newer codes or that code was deprecated for some reason. So I think the maintenance is really important, and actually I think we have something about that in the Quick Start guide about, recommended practices. So if people have questions about that, they should look at the Quick Start guide.
Yeah, I got my voice back. So the other thing is there are tables in the distribution that highlight what's got changed at different levels depending on what you care about. And there's also tables where, say, one test was deprecated and with pointers to the right test for it. So, yes, they should. I agree with Swapna 100%.
Okay, great. And this is a question, I don't know if this is, for Clem and Swapna and or, maybe, I think probably for you guys. Question from the audience about how to represent test panel results over time using charts and graphs. I don't know if there's like an easy answer to this, but, if you guys can answer it, but this is one of the questions. I don't know who wants to take it.
What we did, when I was in Indiana 14, 15 years ago, so the panels were presented in the flow sheet with a title line saying what the panel was, and then the children all listed below. Now, there are panels that overlap. In that case, we'd merge them into one title line with the other children mixed together, either in sort of the physical order when such is kind of imprinted in positions like is true, I think, for the CBC, but otherwise in alphabetic order.
So, Swapna,
okay.
Sorry, Vishali, I'd like to go out on a limb here. This is Stephen, and say that I think this is really one of the areas where we're going to see tremendous innovation. Once, we free this data and we make it available in FHIR, in CDA, by whatever means, and patients can get a hold of it and app developers can work with it, I think we're going to see tremendous innovation in the display of data, the utility of the data, whether you're talking about presenting it to providers, in the clinic, in the ICU, in the ambulance, whether you're talking about presenting it to patients, in their own apps. There's a lot of opportunity for innovators to work in this space because the EHR developers have done what they can, and it's sorely lacking.
Thanks, Stephen. I think that's a really important point in thinking about the future with regards to FHIR and the potential use of it. One last question. I think we just have on the hour just one point. Someone says, this is Dr. Sudha. We have developed a single-click tool using XML that can check all LOINC maps of a lab. and that would provide inputs as to which codes have changed with the new release. So anyway, I think that's just a shout out to a tool that's out there.
Well, I respond and suggested that sounds great. You know, pass it around or pass out URLs or whatever, so you can, you know, the world can use it.
That's right. Great. Well, thank you, everybody. We're at the hour. Thank you to all our panelists, and thank you to the audience for a great set of questions, and enjoy the rest of the Tech Forum. And there's another session, I should say, on lab interoperability as it relates specifically to public health that is following this. So stay tuned, and if you enjoyed this one or want to dig deeper into the public health aspects of it, please join us for that next session. Thank you. And also there's an evaluation. Oh, go ahead. Sorry.
No, go ahead.
I was just going to say, and also there's an evaluation. Oh, go ahead. Sorry.
No, go ahead.
I was just going to say there's an evaluation service